Now we're getting on to another exciting part of the day. The time where we get to learn all about things coming down the pipeline for FSHD therapy development. To share about all the drug development and clinical trials, I'd like to invite Dr. Hans Lochmuller back up to speak about that. After Dr. Lochmuller, we have a guest speaker from Avidity, Dr. Nchienzia, who will update us all on the progress of the FORTITUDE clinical trial for FSHD. And then Amanda Hill, our very own, will moderate a discussion on drug development with Drs. Lochmuller, Nchienzia, and Corngat right afterwards. So, pictures.
Thanks so much. So that's going to be a little bit of a visual tour to all the trials that we are aware of, and the slides that was actually kindly provided to me by the FSH Society. So if there's any mistakes, it's good to bring them. So we already talked a little bit about this. So this is branding the different therapeutic programs in the pathophysiology, which we said starts with the gene. What can we do about this? Then goes to the RNA, to DUX4 as the culprit and the inflammation, everything else, and at the very end, maybe the muscle stem cells for repair to hurt. And at the lower part of that slide, you see the different phases of clinical trials, from preclinical, when it's not in humans, when it's in mice and cells, and then phase 1, 2, 3, and we'll come to that what that means. And the logos of the different companies are assigned to both what sort of mechanism they sort of target, right? So on the very left, you take, for instance, epiCRISPR, so this is on the side of the gene, and there in phase 1. It's basically mapping the whole sort of landscape of clinical trial development to where these things act and at what phase of development they are. But let's take a step back. So what phases do we know for clinical research? Well, there's the preclinical research where labs are involved that look at the disease mechanism and animal models, etc. Then it goes into clinical research, and that's not always starting necessarily with giving people a drug. It's also understanding how does the condition progress, so-called natural history studies, for instance, or figuring out what the biomarkers are and how to assess these things. And then there's the clinical trials, and then there's something after the clinical trials as well. So more specifically about the clinical trials, so why do we call them phase 1, 2, and 3? Well, it's not necessarily the case that all medications have to go through all 3 phases, or sometimes there's only one phase 2 trial, there might be even 2 or 3 for the same product. And the reason why they're called 1, 2, 3 is because they address different questions. So phase 1 is really just about safety in the first place. Does that drug, if we give it to humans, any harm? And that's often even done in healthy volunteers, not even in patients. Phase 2 is then in patients and includes a relatively small number of patients. And it's more about how often do we need to give this drug? What dose of that drug do we need to give? Is it safe for patients, of course, so that sort of longer term? And does it actually hit the target? Does it, for instance, reduce DUX4? That's what we're going to see. It's not yet about, does it actually help people? And that is then that's addressed in phase 3. Does it have a clinical benefit? Does it make a difference for people's health? And that's our obviously the largest trials. They go for a longer period of time, and they're of course also the most expensive. And in each of these phases, a drug can of course fail, or meaning we find out that something doesn't work, I don't actually like the word trial fails. The trial usually just answers a research question. And the question can be yes or no, but there's an answer, right? If you know it's a no, well, we have an answer, it's not failed. I think the failed trials are the ones that are badly designed or that don't have an answer because then we don't know what to do, right? So anyway, so let's go now to the trials. And the first one is actually one that's finished. And it did show that this drug is probably not working. Nonetheless, I think it was a very important and a pioneering trial because it was the first drug that in FSH went all the way from phase 1 to phase 3. And what was this drug trying to do? So it was an oral drug, so a pill basically, called Lochmuller. And that did not work on the gene or on the RNA, worked on DUX4, on the protein, reduced the inflammation, etc. And it looked pretty promising even in the phase 2 trial. So, but it did not show efficacy in the phase 3 trial, unfortunately. And the reason was that in the primary endpoint, so the primary endpoint was being considered the most important to show that a drug is efficacious. There was the reachable workspace test. There was no difference between the placebo and the drug. In fact, both of them showed stabilization or slight improvement rather than what you would have expected from the placebo that is actually getting worse. And this is still not 100% clear why that was the case. And some people still think maybe there is something about Lochmuller that we don't fully understand. But at least in Europe and North America, this drug is not being further pursued. Coming to the next trial, Roche maneuver. So this is a trial that is not run here in Canada, so I don't have any direct experience with it. By the way, Ottawa was part of the clinical trial that I should have mentioned. This trial is a myostatin inhibitor. So what is myostatin? Myostatin is a small molecule that regulates the size of the muscle. So you can imagine everybody has smaller muscles, bigger muscles. There are some people that have very big muscles because they train a lot, etc. But there's the body tries to figure out how big muscle you actually need, and therefore it regulates the muscle size. And one of the molecules that the body is using is called myostatin, and that keeps the muscle small. So the idea is, and it's not a new idea, it was a previous trial with a similar drug, to reduce that molecule that keeps the muscle small, and by that they help people grow bigger muscle. So these are so-called myostatin inhibitors. And Roche has one in the clinical trial currently that they think is superior to previous myostatin inhibitors that didn't make it. It would obviously not be exclusive or specific just to FSH. It could potentially work in other forms of muscular dystrophy as well. This is the study design. It's currently in phase 2, and as I said, it's not run in Canada. So unless someone is participating in the States, it's not relevant until we have a result. The next study is also a study where Roche is involved in some respect, but in that study, they only provide the medication. They are not the sponsors of the trial. The sponsor is actually an academic hospital in France, in Nice. And this drug is what we call an interleukin-6 inhibitor. So this is a drug that is already in clinical use for another indication for an inflammatory brain disease. And it's also called satralizumab. So it's available in Canada for this indication. But it's not indicated for FSH at this point. And what our colleagues in France, Dr. Sacconi, found that IL-6, this inflammatory molecule, is increased both in muscle, but also in blood of patients with FSH, and thinking that inflammation is sort of an important part of the downstream pathology of FSH, reducing this inflammatory molecule would have benefit. So this is a phase 2 trial as well. There's a total of 40 participants. 11 of those are in Ottawa. The remaining ones are in Nice, and we are fully enrolled for this trial. The double-blind phase runs per year. So by early next year, we should be having those results and see if this trial goes forward. I should say this is a drug that's being given through subcutaneous injection of this molecule every 4 weeks. So it's similar like people use their insulin pens. So it's relatively easy in terms of administration, can be done by patients themselves. They don't need to go to the hospital. Next trial will be covered by the next speaker. So I'm going to give you very little information other than saying that this is a drug that we also run here in Canada, in Ottawa. It's a phase 2 trial at this point with an antisense oligonucleotide that targets DUX4. And the study is a phase 2 trial and is fully enrolled at this point, and look forward to hear more about this. It's an international trial, but Ottawa is the Canadian site, and hopefully things move along as expected. We're going to have a phase 3 trial as well coming up, where the other Canadian sites will be involved as well. And then I also have a couple of trials again that are not available in Canada. One is Arrowhead. Again, it's an antisense molecule, similar to what we said about Avidity. It's a phase 1/2 trial, so relatively early, and it's only run in New Zealand, Australia, and Thailand. And I don't have a lot of specific information about it. And then I think I already heard people asking about this one, CRISPR. So this is an interesting one. So this is not targeting DUX4 or the DUX4 RNA. This is sort of targeting what I explained before, the switch, right? So to make sure it's switched off. So it's trying with a CRISPR to methylate that repeat sequence, and therefore stop DUX4 being made in the first place. CRISPR is known as a molecule that modifies genes, and the CRISPRs that are often thought of are the ones that actually cut the DNA or change the sequence. This one doesn't do something else. It's basically just binding at the right spot and then methylates that region. So this is also a very early trial, phase 1/2. It would be a one-shot, basically, therapy, and it's currently only available in New Zealand. And we'll see where that is going. So there's other programs, and we've heard about Zatellos, and maybe there's other companies coming up that are currently still preclinical. So that landscape is ever changing, and that's great that so many things are available, being looked at. And from a Canadian perspective, as Victoria said, we have a website with NMD4C and NBC that sort of lists these opportunities where you could look things up about what's going on and what's available in Canada, because looking up clinicaltrials.gov can sometimes be a bit challenging because there's just so much information there, and not all of it may be relevant and explained well enough. But what I should also say, besides those clinical trial opportunities, so clinical trials, well, either they may not all be here in Canada, or even may not be close goes to a site that runs a trial or not have the time, or maybe not even interested to be the guinea pig for a new medication that nobody knows at that point what it does, right? But you may still want to contribute to research, and you can do that. Everyone can do that. And one of these opportunities is the MOVE or the MOVE Plus studies. So in these studies, you actually just need to come to site and give a blood sample and have some imaging done and a few assessments. So it's not as intense in terms of scheduling, etc. And you can, through that, contribute to research into our understanding how these outcome measures work and how the progression is, etc. So that's also hugely important. And the initiative that's done a really fantastic job in that is that network, the FSHD CTRN. The image that's Dr. Stadland, who's chairing this initiative, that is now encompassing a lot of sites in North America, some in Europe, as well as Montreal, Ottawa, and Calgary as well. So this is another way, an important way to contribute to the research. So that summary slide actually is not 100% correct anymore, because currently the Avidity clinical trial, as far as I know, the phase 2 is fully enrolled. Roche maneuvers in the States, so actually we are clear at this time right now, there isn't a clinical trial with a medication actively recruiting in Canada. That was different 2 weeks ago and maybe different a few weeks from now. So it's always changing, but you came to me and said, I want to enroll with you in a trial tomorrow. I have to say, no, we don't have a trial open right now. But it's worthwhile, if you're interested, to actually get known, sign up to the CDR, sign up to research lists, etc., make yourself known. And then when the next opportunity comes along that is of interest, then of course you will be considered. For now, I would encourage everyone to think about that they want to get enrolled in MOVE Plus and contribute this way. And this is open for recruitment at several sites. All right, I think that was the whistle tours of different clinical trials. A lot of information very quickly, and I don't know whether we for people who are interested to provide this slide set. Yeah, we can. That's excellent. Thank you very much for your attention.
I'm gonna have Henry come up. Yeah.
Thanks, Hans.
All
Good
right,
job.
thank you, Hans. All right, we're gonna now have our Henry Nchienzia from Avidity. Please come up. We're gonna talk a little bit about fortitude. Just a quick note about Dr. Nchienzia. He joined Avidity a year ago as the senior medical science liaison, working to establish Avidity among the medical community. And I will say I got to meet Dr. Nchienzia his first week on the job in Austin, Texas.
Thank you, Beth. And that's been a long journey.
Yeah, well.
Which has been amazing. Well, thank you, FSHD, for having us today. And as Dr. Lochmuller did mention, this study is fully enrolled. And right now we're thinking about the global phase 3 study, which will be announcing shortly. So the results I'm going to share with you, it's a result that was presented last year. So just stay tuned, because we have another result coming in June 13th. So a month from now, we'll have more data to share. So this is just a data from last year. We're a publicly traded company, so I have to show the slide. And please, as the previous speakers have said, don't try to read them because you might have crossed eyes. As a company, I believe this is our platform. So we are in DM1, which we have trials here in Canada too. We are also in Duchenne. This is phase 1/2. And then we are in FSHD, and the target for FSHD is known as AOC 1020 or the BRAS for short form, because honestly, the parent name is so long, we just call it the BRAS. And right now we are in phase 1/2, and as Dr. Lochmuller said, phase 1/2, we are trying to see how well and how safe this drug is. And for phase 3, we'll look at the bigger question of, is there an effect of the drug? So, I really want to thank Dr. Lochmuller again for going through the pathophysiology of the disease. But what we are targeting right now for us, we are looking at DOX4. So, let me see that. Okay, great. So this is what DOX4 will do in a disease condition, right? We can see that based on the number of repeats, DOX4 will then result in the transcription of factors that will lead to cell death, immune response, as well as oxidative stress. That is all what will lead to your progression of FSHD. What we are proposing as a company with our molecule, which is known as Dobrax, we have a small difference RNA. So what that would do is it would interact with DOX4 and kind of destroy DOX4. So when that destruction happens, we then halt everything that happens downstream. For example, the cell death as well as oxidative stress that will result in progression of FSHD. So that is just the thought behind the molecule that we're developing in clinic. So this is just high-level information about the trial. It's a phase 1/2, so it's randomized placebo-controlled, which means that you can either be in a placebo arm or you could be in an active drug arm. And we had an age of 18 to 65. Good news, I'm going to say this, for our global phase 3 trial, we're dropping down the age to 16. So we enrolled from 16 to 65. And we had a couple of biopsies for these, and then it was ascending dose. So in the phase 1/2 trial, what happens is we start with the first dose, the lower dose, and then we progressively up the dosage. So that's why you see it says cohort A. Our first dose was 1 milligram per kilogram, and then we then up the dose to 2 milligrams per kilogram. Phase 1/2 we're looking at safety and tolerability. That's just to see how safe this drug is in humans. And then we had some exploratory objectives. So we're trying to see the biomarkers of this drug, and also looking at some measures of strength. For example, we're looking at muscle strength, muscle function, as well as muscle composition. And we're always interested in the patient and clinician outcome of the drug and the trial. So these are the demographics, pretty similar, but then what happened is you realize that, as I said, this is the data cut that was done last year. This is just for 12 patients. We had 8 patients in the active arm of 2 milligrams per kilogram, and then we had 4 patients that were in the placebo arm, which means the arm that did not receive any drug. So if we can compare the dynamics or demographics, looks kind of similar in terms of the age and clothes and the genetic disease, as well as the different repeats. We also use a visual workspace, which is looking at different quadrants of your arm extension, and we are interested in the muscle testing. So right now, the drug is safe and is well tolerated. You will see that there's a 4 milligram per kilogram, right? This is an extension of the study. So what we did is we're collecting data as we go along, and we are very confident to share that right now there is a serious adverse event. An adverse event is any occurrence of anything that happens within the trial, right? So we're trying to make sure that every of such events are monitored. Right now, we don't have any of those events, and nobody in the study has discontinued because of any unforeseen circumstances. And so we wanted to make sure that if we are moving on with the study, we actually have drug in the muscle. So this is about cells taking just to make sure that if we are continuing the study, we are confident that there is drug in the muscle. So on the y-axis is a small difference RNA, and that is a molecule that we deliver to the muscle to target DOX4. And then we could see the dose is 2 milligrams per kilogram. And here we are showing that at any given time in the muscle, we have drug between 1 and 10 nanomolar. So this is like an internal control for us to make sure that we are delivering muscle or drug to the muscle. This is an interesting graph. Please don't try to understand anything about explain very, very quickly. So at the top panel, we're seeing, if we look at the top panel to my left right there, this is baseline. So the red that you're seeing right now, in a normal condition, everything that is red should have been blue. Right? But under FSHD, with DOX4 activated, we are seeing that it is red. Mind you, it is individual patients. That's why, and in the model we heard about, there is heterogeneity in this disease. We can see that one patient has a lot of red, another patient has some level of red, but then there's some level of blue too. And if you look at the panel below, these are genes that are supposed to be red, but then because of FSHD, they are now blue. And yet again, we are seeing the heterogeneity of the disease. But then what happens is once you give these patients the drugs, and these are patients that receive active drug, we are realizing that in the top panel, there is a switch now. We're seeing the genes that were supposed to be blue, which is downregulated, have now been downregulated. And likewise, on the bottom below, we're seeing that the genes that were supposed to be upregulated, but because of DOX4 were downregulated, after we administer the drugs, we are now realizing that those genes are upregulated. And these are all individual patients that were part of the active arm that were showing the biomarker. And we also, we heard in the morning that measuring DOX4 is very tricky because the expression is very variable. So what we did was that we looked at some genes that are regulated by DOX4 that we can measure. The ReDOX4 panel as well as the 41 gene panel, these are established panels of genes that we know are regulated by DOX4. As a company, we are also developing a panel in-house, also looking at the regulation of DOX4. But what I just want to drive attention to is irrespective of the panel that you're looking at, for those patients that were on placebo, that means no active drug, compared to the patients that were on active drug, we can appreciate that there is a consistency of reduction of those genes that are regulated by DOX4 across the panels. So this is just also give us an idea of we are targeting DOX4 and we are seeing some improvement in these patients. And everybody was on 2 milligrams per kilogram of drug. Likewise, we are also looking at circulating biomarkers. We are also developing a biomarker in-house, but then we also looked at creatine kinase, which we know is released when muscle is damaged. So what we are seeing here is, just like what we observe in creatine kinase and an internal biomarker, those patients in orange that are active drug and the patients in black are those that are placebo arm, which means no drug. We can appreciate that while the patients are no drug, they have the circulating biomarker or creatine kinase maintained. The patients on active drug have a reduction of the circulating biomarker as well as the creatine kinase, which is kind of a hint of muscle integrity or muscle restoration. For exploratory efficacy, I talked about reachable workspace. So we looked at different quadrants. So we can see for Q1 or Q3 or Q5 or Q4, what we're observing is that compared to the people on the placebo, the patients are on the placebo arm, in this box plot, we can see that if you are trending to the left, it means there's a decline or there's a change. And if you're trending towards the right, it means that there is some sort of an improvement. And we can appreciate that there is a trend here with the patients that were in the active arm, the 2 milligrams per kilogram, compared to the patients on placebo, that they tend to improve compared to the patients on placebo. So right now, this slide has to be updated because we had the cohort C, that's a biomarker cohort. And as Dr. Lochmuller explained earlier on, to get drug approval, you need to go through your phase 1, 2, and as well as phase 3. But then what we're trying to do right now is with the biomarker cohort that is actually and actively completed, we are looking at initiating conversations with the FDA to see if the biomarker cohort, the results from that is sufficient enough to get to an approval. But then we still need to conduct a phase 3 trial. So the phase 3 trial will be announced sometime this summer, but then it will be a global trial that we have to conduct irrespective of the conversations that we will be having with the regulatory bodies. So right now, we advise everybody to stay connected to clinicaltrial.gov, and please stay plugged in to FSHD because they have a lot of information and they have up-to-date information because we share with them. And please don't forget to conduct or consult with the doctors to get more information. And also please visit our website because we have a lot of press releases coming up. As I mentioned, we will have a data release coming up in June just to share more data to what I just shared with everybody. I know that I think that we don't have to come up again, or we have a Q&A coming up.
Yeah.
So thank you all so much, and thank you for listening.
Okay, so now we'll have Amanda, if you'd like to come up to facilitate our panel discussion, Dr. Korngut. So both Drs. Lochmuller and Korngut are facilitators or PIs on clinical trials, so they're the clinical experts. And then we have our drug company rep. And we have a set of questions that we're that Amanda's going to ask our panel. This actually better. You get your chair. No, Amanda, it's so funny. Yeah, right. Oh, she just needs a microphone. Yeah, you could take this, Amanda, if you want. This one's better.
Yeah, the song.
A lot of questions might be for you.
Yeah, it's me on one.
Okay, all right, okay.
All right, lovely. Okay, so just to kick things off, so we have Dr. Nchienzia, did I say that correctly?
No.
Henry,
Yeah.
lovely, thank you. And then we have Dr. Korngut here to my left, Dr. Lochmuller here to my right. So Henry is here representing Avidity. Dr. Korngut and Lochmuller, maybe you can clarify for us, do you have any, we know there's no active clinical trials right now for Canada actively enrolling, but what clinical trials maybe have you both done at your site recently or other studies relevant for FSHD at your sites recently?
Yeah, so our site's been very active in neuromuscular clinical trials for about 15 years. And I think we've done just over 100 different trials across various different diseases. But FSHD, of course, is new in terms of the clinical trials that have been emerging. The first FSHD trial we did was the Acceleron clinical trial, which was now probably about 5 years ago, I think. And so we then did the recent Fulcrum clinical trial. I think we had 5 or 6 patients participating for that. And now we're waiting for the next trial. And of course, Avidity is the next trial, their phase 3 that's coming out, in the near future.
Yeah, I think I mentioned already, so we've been involved in Fulcrum phase 2 and 3, in Avidity, as Henry described, and the reinforced trial, and we also do the plus natural history study for FSH.
Great, thank you. So just could get a lay of the land here. So the first, I've got a list of questions that you all actually asked before this event. So we'll get through hopefully a lot of these today. So to start off, we'll ask for Henry and what types of patients would be good candidates for the Avidity drug, and then maybe doctors Korngut and Lochmuller can comment as well on some of the other studies.
Yeah, thank you, Amanda. So right now for our study, we have patients that there is confirmation of the disease, and then have some muscle weakness as well as upper weakness or lower body weakness. We understand that there is need for more trials, and that one trial alone would not, it's not a one size fit all, and definitely we encourage everybody to either plug into new trials or new study, because that's how we still help to move the needle forward in terms of research. for us right now, criteria, and we're thinking that in phase 3, we might open up the criteria a little bit more to get more patients get into the study.
Yeah, so maybe I can comment on something that I think is an important issue that we'll all see come up, that we've seen in other conditions. As we heard earlier today, every individual with FSHD is very unique. And I think we all recognize that. When clinical trials are conducted, we use two groups. So we use a treatment group with the drug and then a placebo group. And we don't know the individual with the disease doesn't know which group they're in. Ideally, the groups look exactly the same. And the only difference between the two groups is one group has the drug and one group does not. Of course, that's not reality. We don't have, we don't clone people to do, clinical trials. And so, but as you introduce unique individuals, you can imagine those two groups are always a little bit different. It's our job to make sure they're as similar as possible, because the risk is if the groups look too different at the end of the trial, is that the effect that we see isn't due to the drug. It's actually due to the difference just in the inherent individuals in the two groups. And the challenge with that is it can give us a false positive where it says, wow, this drug really does something when it doesn't. Or it can give us a false negative where there's a drug that works that we think does not. And so you can imagine there's a lot of kind of thinking that goes around what are the criteria and what kind of individuals should be permitted into this trial. And importantly, that's the reason. It's not that these companies or researchers are trying to exclude people, because it feels like that. But it's because we're all trying to get an answer, as frustrating as that is. The challenge is that then later compounded by the fact that reimbursement may just be for those individuals who were able to qualify, right? And so there's a lot at stake, but we've seen this in other diseases where the company and the researchers make it too broad, and then it's a negative trial, even though everybody thought the drug would work. And so these are the risks of the whole process.
Yeah, so if I just may add to that, so besides that aspect that there's inclusion and exclusion criteria aim to have a fairly homogeneous group, they serve two other purposes as well. I think one is to actually make sure only those participants get included that are considered safe, meaning if somebody has severe heart disease or severe kidney disease, and that depends a little bit on what the drug is actually doing, then they may not be allowed to be in the trial for that reason, because obviously that's the first thing we want to avoid, harming anyone. And the other aspect is so to figure out whether the drug works or not, we have those outcome measures, and that there are strength tests, walking tests, MRIs, whatever. So you want to make sure that the people who enroll in the trials can make those tests to start with, and secondly, that these tests show something that can change over time. So this was, for instance, an experience with some of the trials that people had to fulfill certain criteria on their MRIs, and if they didn't fulfill these criteria, then unfortunately we had to tell them you cannot proceed with the trial. But the reason was that, well, that was an important endpoint, and if there's no chance to see what we want to see or the change that is expected, then it obviously doesn't make sense for them to proceed in the trial.
Thank you. And this brings up for me a question that we get a lot. When you look at the trial criteria, this inclusion exclusion criteria, there's a specific age range. A lot of times people need to be ambulatory. That's very common for the studies right now. So a really great question that came in is, what is needed in terms of research, policy, and advocacy to have clinical trials for younger patients under 18, under 16, older patients over 65, over 70, and the people that are non-ambulatory who are currently excluded from trials?
Yeah, I think I think we all want to see, as much access to these trials, and we certainly want to learn as much as possible to understand. I mean, it makes sense to treat somebody who has a condition that they're born with as early as possible, right? I think that's logical. You know, the FDA in the US and Health Canada, you know, they do like to see if it's a condition that spans childhood into adulthood, that it's shown safe in adults first. Now you can debate what adulthood means. Is it 18? Is it 16? Is it 50? You know, so I think there's some room for discussion there. I think as individuals get older, I think some of the concern is there's other medical issues that arise, and then there's at a certain point, virtually all neuromuscular conditions get to the point that the treatments may not work, even if they do work. And so there's a lot of questions. I think importantly, it's not always one trial or two trials. There's often more trials that are done over time in different age groups and different subpopulations of the community that we have to consider. But I think, you know, we've already had one kind of warning to my earlier point that the Fulcrum clinical trial, which was just reported as a negative trial, in that trial, we expect the placebo group to progress. So it was individuals with FSHD over 48 weeks. And so it was expected there would be a decline in motor function, and there wasn't. And so it's really hard for a drug in a disease like FSHD to show benefit if the placebo group's not getting worse. And so that was a bit of an alert to the community to sort of say, okay, we need to go more narrow and really make sure we're finding individuals for these trials that are going to progress in that time period. Because otherwise, again, we're going to have a negative trial, again, even if the drug works. And so that, just to give you a sense of kind of some of the debates in the background that kind of go on, because it's frustrating, but in the end, we you know, we need results, right? We need progress.
And I just want to add that, you know, natural history studies are very important because that is what informs us of our endpoints. Because then we can look at the progression of the disease and inform us what endpoints as a company we could use for our trials. So please, natural history studies are very important. Thank you.
Yeah, and just as an addition, so of course, FSH is a relatively rare disease, and well, there is of course then the possibility if you run out of patients that sort of fit those sort of mainstream criteria to go to other sites in other countries, etc. But that has some risk and limitations as well. So I think what we've seen in other conditions is if there is a lot of competition around a certain age group or a certain functional group, that then there's thinking about the researchers and the companies, should we try this in a separate group that maybe cannot do the walking test, but still may benefit for the arms or something like this. So I think that's entirely possible. The other option is, of course, if you don't aim to actually show efficacy in these groups that are a little bit more diverse or a bit more outliers in terms of their functional age or etc., you can at least sort of see what the safety profile is in this population, and that may actually help you then with the approval later on. So there's some ways people that don't sort of fit that sort of standard sort of profile get included in the research. And of course, there is also incentives actually once it's shown efficacious in adults to test the drug in kids. The companies get extra protection for their patents and how long they have exclusivity, etc., or get other incentives for that. So, but one, of course, important thing is that in the natural history studies, in those studies where we want to understand the biomarkers and the outcome measures, etc., these other groups are also included, and I think that's the case for MOVE+. And there's I think a pediatric study is being planned as sort of similar to MOVE to extend, and Canadian sites want to participate in that as well.
Great, thank you. And another way that we can get at some of these questions, these hard questions that you have brought up, is through compassionate use or extended access programs. So we have a question here is, you know, will there be a compassionate use or extended access program, Avidity, of your therapy for people who are not currently eligible? And then I'll expand that maybe to doctors Lochmuller and Korngut, I'm gonna try to combine your last names here. Maybe you can comment on what you've seen in other disease areas as well.
So, in Canada right now, we do not have the infrastructure for, vaccinated vaccines use. So we're trying to just focus on, our efforts right now to have conversations with the FDA to see how well we could get this drug approved with what we have. But we do not have the structure in Canada right now for, vaccinated vaccines. We can deal once to add on to that, or we can see guys want to say.
I like it. We'll help you as much as we can.
Yeah, so only one possibility is already true for some of these programs, that once people finish the double-blind phase and sort of contribute to the research, that they're allowed to stay on the drug, and that's true for both the people who were on the placebo as well as the people who were on the drug. And that's what's called an open label extension study. Sometimes this is part of the main protocol to start with, and then they don't even need to sign up again. Sometimes this comes as a separate, an extra study. And what I've seen from some companies, I'm just suggesting something for you, that once these open label extension studies are open, that they would actually offer that some people could newly enroll into the open label extension study, even so they haven't been on the main study. So that's another option. And yeah, so there's of course the question of compassionate use or expanded access, etc. Often these things only come into play once there was actually a successful positive phase 3 trial, and that's not the case for any of these drugs yet. So this is to bridge a delay, because it takes a bit longer for our regulators to approve, or because they haven't made up their mind, or there's some other specific situations for that. But I think that's a bit too early for those drugs that are currently being investigated.
I'll just comment quickly in other diseases. So we've certainly had this come up in other conditions. So in ALS, for example, it's important to remember when there's a drug in phase 2, if we look at the statistics from a successful, promising phase 2 into a phase 3, it's about 50% success. So meaning 50% of clinical trials will then show benefit and 50% will not show benefit. It's actually about 52%, I think, of all phase 3s. And so the important thing to recognize, there's still a lot of risk that the treatment does not work. And there's often side effects and things to consider. I'm not speaking about Avidity specifically, but just overall. In ALS, there was a very striking result in a phase 2 trial. And of course, that's a terminal disease, rapidly progressive. So the Health Canada allowed us to place patients on this drug while we were waiting the results of the phase 3 in the US. This drug had considerable side effects. Something like 30% of patients ended up having to stop the drug because of abdominal discomfort and diarrhea and things like this. And then the phase 3 was negative, so we had to pull everybody off. And so you know, it's a difficult situation, right? Because we just don't know. Often the special access programs come into play when FDA makes an approval. Most companies go to the US first to get regulatory approval. It's a much bigger market, 10 times or even larger. And then Canada, the average delay is about 2 years for Health Canada to make the approval. So what Health Canada will allow is to then, if FDA is approved, they can do a special access program in Canada for that bridging period between the approval. But they don't always do that, right? That's up to the regulator and the company, et cetera. And there's always lots of different considerations. But the point is, you know, the science matters, and we need to know if this is helpful, because there's always some side effect. There's always some risk. There's not to mention there's hope, and there's time and effort. And so we'll have to see what happens, right, with these different drugs.
Great, thank you. Let's see, I'll switch gears a little bit. What kind of a difference would it make to clinical trial design? So thinking about that inclusion exclusion criteria, how many patients you need, how long the trial takes. Once there's a validated MRI or blood biomarker outcome measure, how will that change things?
Come on.
Well, I think if you have a biomarker, the benefit is you can see if the drug is sort of having some kind of biological effect in an individual. And so what that allows you to do is to sort of understand who's responding and who's not responding. And then often from the regulatory perspective, you can better predict who's going to have a positive response to a treatment. So usually it results in shorter trials, less patients, ideally. But also depends on the biomarker, how effective is it, how convincing is it, and all of these things. And so, if you have high blood pressure and you take a pill and your blood pressure comes to normal, that's a great biomarker to suggest that pill is going to prevent complications of high blood pressure. That's probably sort of the most classic example, although it's never quite as clean cut in the neuromuscular system, right?
Yeah, I totally agree. So it would be hugely helpful to cut down the duration of the trials and possibly with the number of patients or stratify patients better to understand who might benefit and who's not. So for the research, it would definitely be a bonus. Whether it helps for the approval, that's a different story, right? So far it's been that EMA, Europe, and Health Canada would not necessarily go for a biomarker to base their approval on. They would still want to make sure there is clinical benefit demonstrated in the phase 3 trial. While FDA has sort of moved a bit towards giving approval, or at least preliminary approvals, conditional approvals, whatever, based on biomarker results.
And just to add to that's why we have a biomarker cohort, which we just completed, and we're thinking of using these results to initiate conversations with the FDA to an approval path. But then we still need to conduct a phase 3 trial to get definite results on how this therapy is in time.
Great, I'm going to try to squeeze in 2 questions here. The first one is, here we go. Will your therapy or other therapies being developed be standalone treatments, or would they eventually be part of a cocktail of therapies?
Right now, we are investigating the rats as a standalone therapy. But then we heard in the morning about this multi-system disease. So while we are investigating our therapy as a standalone, I would want to turn it over to physicians to maybe just walk us through what the thought is as patient management in clinic.
So yeah, so obviously theoretically one could envisage that people have 2 or more of those therapies if they go along with each other. First of all, right, that they don't bring up any sort of additional cumulative or whatever side effects. So if they're safe in combination, and secondly, if they sort of attack different parts of the pathogenesis, right? So you could imagine to combine an anti-inflammatory with something that's reducing the DUX4 RNA or something like that, right? Or throw in something that helps with regeneration, etc. In the short term, I would say that's probably not very realistic because I expect these drugs to be expensive in the first place. Well, first of all, they need to be approved, and then when they get approved in combination and will they get paid for in combination, these are different questions. So I think we, on the long run, I can imagine, and in SMA, where we have 3 approved therapies by now, in a way that's even happening because some kids do get the gene therapy first, and if then that's a one-shot therapy, even so it's very expensive, and then later on, some of them go on some antisense as well. So I would exclude similar things happening for FSH, but I think not for the near future. So be happy to have one drug approved for starters.
Hopefully we'll get there soon. All right, so the final question, maybe we can each provide a quick response to is, what can the people in this room who are living with FSHD do to be ready for clinical trials when they do come to Canada next?
I would say, just stay in touch with the providers, log into FSHD 360. I mean, there's a great resource for you too. Clinical trials go also helps, but then just stay informed with the providers and, stay productive.
Yeah, I think we've heard it today. I think somebody mentioned, it's your FSHD. You know it best. These are your decisions to make. And I think being engaged, being organized, working together, I think is key. As clinicians, we don't get very far without working with the community, and vice versa when it comes to clinical trials, et cetera. So I think it's time to kind of all come together, and you're seeing the opportunities to do that. Because there's a lot of complex things that are going to happen before everybody's really happy about having access to a therapy that's transforming lives, which is what we want. That's the goal here. We don't want just the therapy. We want something that really has a huge impact on quality of life and function. But as we've learned from other diseases, often that requires a lot of advocacy. Often that requires a lot of getting together to collect data, to make arguments to health systems, to et cetera. And so I think it starts with today, like this kind of session, and then trusting each other, working together, learning from each other. It has a huge impact. Communities that don't do that, things just don't get very far.
Wonderful, thank you.
On an individual level, I would say, well, for sure, stay informed about what's going on. Sign up to the CER, and also, well, look after yourself and stay as healthy as possible, right? So do the exercise and everything else that you heard about, because that's obviously you want to get into a trial at your best possible, right? And I think on a sort of community or group level, I would say, besides sort of supporting the sites and the research, etc., there's also this piece about policy as a rare disease. If you get together as a group and advocate to policymakers, maybe in some instances together with other groups like Muscular Dystrophy Canada, that can go a long way, so that at the end of the day, not only more research is happening, but also then the drugs that hopefully will get approved and people get access to them, and the hospitals are in a position to provide you with these drugs. So there's lots of things that need to come together, and as clinicians and researchers or industry, we are obviously the people who should be doing the work, but we're always sort of seen as self-serving. If we go to politicians and say, you should give us more money for this research on muscular dystrophy or on FSH, I think you as a patient group have a much better starting point there with the politicians.
It's a group effort. We're all
It's a group
yes,
effort.
wonderful. Well, thank you all so much for joining us today.